opening the door wide to new possibilities, hope of real breakthroughs and understanding more big ideas than we could have ever imagined. These are the biggest grants we've ever been able to offer. We're going to come out of this with some breakthrough ideas.
Focused solely on neuroendocrine tumors. You are not alone. We are all with you on the board.
One of the things we're very proud of, in addition to the fact that we actually have funded the trial, that the so-called antagonist trial that Dr. Reedy talked about with her colleague, Dr. Weber. One of the things we're also very proud of is the fact that we bring together multiple institutions for these education programs. So you've heard from, and we'll hear a lot more from the Montefiore team, you've heard from Sloan Kettering, both alum and current leaders there. You'll hear in a moment from Dr. Metz again from the University of Pennsylvania. It also bespeaks the kind of collaboration you've heard throughout those days. So you have a lot of resources in this room and beyond. Use them. So we're going to talk now a little bit about the future and why we're so excited about the future in terms of research. And it goes to the work that we do at the Neuroendocrine Tumor Research Foundation. Just a couple of quick words about that, then we're going to introduce our Director of Research, who will introduce one of the very exciting projects in immunotherapy that we're about now, and then we're going to get to lunch. So we are a research foundation that I mentioned earlier today was founded by a neuroendocrine patient who, after being diagnosed some 16 years ago, looked around and found very little was being done, very little funding for research, very little on clinicaltrials.gov, very little funding available, very little to read and learn about, but most importantly, not a real focus on cure. And so she pulled together a dynamic group of people who set themselves on the goal of developing an organization that was totally focused on raising private funds in a nonprofit platform to drive the research that could result not only in dramatically better treatments, but also for cure, and to also do the kinds of programs here we do today to support our patients. Over the first 10 years of our existence, we've, mostly through the help of a lot of those foundations and individuals, and some of you in the room here today, small donations, somewhat larger donations, we funded about $12 million in research. There are lists of all of the projects that have been founded on our table in the atrium. And the key thing is that we stay focused not only on the research, but also on that urgency that we bring to the mission of finding cures. We have a small board of trustees. Every one of them is either a patient or a family member of a patient or who's lost a patient in their family. They keep us very riveted and focused on the fact that we want to do everything we can to get as quickly as we can to the goal of cure, and that's what drives all of our decisions. But what really is the key input to those decisions is an incredible board of scientific advisors who, for now more than 10 years, some of them, have been giving us their expertise, their time, their energy, without getting paid a penny, because they are totally committed to the same goal. And you'll hear more about that from Lauren in a second. We take a very collaborative approach to our work. We bring those researchers, not only our advisors, but people like Dr. Reedy, Dr. Metz, and others you'll hear from today, and many who've been alluded to in the presentations today. We bring them together every year. Dr. Woolen's always at these meetings along with it, because we believe very much that, especially in a small space, getting researchers to share data even before it's published, which can take a long time sometime, to really learn from each other, to have the kind of collegial interaction that allows some of the collaborations that you heard from Dr. Woolen, from Dr. Reedy, you'll hear more about in a minute. That's going to accelerate process by itself. So we're very committed to that. And of course, as I said before, we're really committed to making the kinds of grants that allow us, through others, NET foundations, the AACRs, the American Association of Cancer Research, opening the door wide to new possibilities for bringing in excellent young investigators, but also people who are experts in other fields, which is one of the things you'll hear about in immunotherapy, to focus their expertise and energy on helping us achieve our goal of cure for NETs. Our big news at the end of last year, the beginning of this year, was the fact that after 10 years and $12 million of funding of research, we got a huge, wonderful gift from the Margie and Robert E. Peterson Foundation at the end of last year. It's $15 million over 3 years. We got $5 million this year. Lauren's going to talk a little bit more about what's happening on some of these fronts. But it is so exciting. Mr. Peterson, Robert E. Peterson, died from a neuroendocrine tumor some years ago. He was an automobile publisher, and so you'll hear a lot of allusions to acceleration and putting our foot on the pedal, but it's really all about what we, in dialogue with that foundation, decided is really going to be the way to not only accelerate but dramatically change the landscape of the research that's going on. A lot going on, as you heard. This has been a very exciting time. We are committed to making sure a lot more goes on in the months and years ahead. And so this is leading to some unprecedented collaborations for us with the European group, with the North American group you heard earlier, with the international group that I head up on a number of fronts. The headline and the central focus of our efforts with them, Lauren will talk more about this, is the Accelerator Rfp. We call these Accelerator grants because, appealing to the Petersons in particular, we see these are the biggest grants we've ever been able to offer to the neuroendocrine tumor community. $1.2 million over 4 years. We said to the world, and we reached out in Europe, in North America, and around the globe, we wanted the biggest ideas, the best ideas, and we wanted teams, multi-institutional collaborations, teams working within and outside of multiple investigators that would give us the best hope of real breakthroughs in understanding what makes these tumors tick, how to stop them from growing, eventually how to find cures. We have had an amazing response to this RFP, more big ideas than we could have ever imagined. For the first time, this international neuroendocrine tumor community is focusing on one process, one set of grants, one super sophisticated scientific review process. Our hope is we are going to come out of this with some really potentially breakthrough ideas that are going to accelerate not only the progress of understanding, but also accelerate the progress of better treatments and cures. And we will bring these teams together, however many we're able to fund, so that we continue to accelerate their learning by their sharing of data and insights. We've also in this process made some grants. You heard from Dr. Metz. This is the first time we did this, funding that investigator at the North American Neuroendocrine Tumor Society. Interestingly, the breakthrough result, finding the first mutations in neuroendocrine epigenetic mutations, they called them, in 2011. That was at work at Hopkins, as Dr. Woolen had alluded to earlier. We now have just funded this young investigator at Hopkins again to take the next step in seeing what we can do to take those findings to affecting tumor growth. The Educational Research Foundation has just announced, we asked them to do it by today, their first pilot, which we are funding for nuclear medicine. All of this excitement about Prrt and how we can bring it to the fore, and you'll be seeing a picture of that young gentleman in a moment. Another clinical trial, a small clinical trial. Dr. Reedy said sometimes it takes a small private foundation to do what we're doing at Sloan Kettering with that antagonist Prrt, and to do what this gentleman, Dr. Tom Hope from the University of California, San Francisco, is going to do, to look at a new way of introducing some of that radiation so it only goes to the liver and doesn't go to any other places, because that, for a lot of our patients, is really the key in terms of dealing with their metastatic disease. So we're very excited about the door that the Petersons have opened, but we are committed to going through that door and taking advantage of every opportunity in terms of these proposals. So if we're not, and it's quite likely we're not going to be able to do everything with the Peterson funds, we're going to be looking to our donor community, and certainly would appreciate any support from anybody who you or your network understands has the same commitment, so we can go forward with that. So I'm going to turn it over now to Lauren Erb, who was the first and only for a long time employee of this foundation, the first executive director, and now our director of research, to talk about what our process is and what we're excited about next.
Terrific, thanks. Terrific, thanks, Ron. It's really a pleasure to be here. I think actually my first patient education event that I ever attended was maybe my second week of work back in the early days of the Caring for Carcinoid Foundation. And the very first speaker that I ever saw speak was actually Dr. Warner. It was a patient education meeting put on by Marianne Wahman in New York. So it's really wonderful to be back in New York again after all these years, and to have again Dr. Warner being here. It's really nice and special opportunity for me. So I have the exciting job, I think, of talking a little bit more about how the research process works here at the NETRF. I'm going to be talking about both our research strategy and our portfolio. So on the strategy side, we really do have a strategy at NETRF, and that relies really strongly on the expert advice and guidance that we get from our scientific advisory board. We have a really terrific scientific advisory board of experts not only in neuroendocrine cancer research, but also outside but important related fields, things like immunotherapy, cancer genetics, mouse modeling. All of these areas of interest really help feed into our setting of priorities. And in that same vein of thinking, we've also been known, especially recently, to take a really leadership role in driving a research agenda. So rather than just always going out to the community of researchers and saying, you know, what do you think we should do? We oftentimes actually go out to the research community and say, you know, for example, we think that mouse cancer modeling is very important. We think that epigenetics is very important in this disease, and we want you to begin thinking about how you might study these processes and then bring them back to neuroendocrine tumors. And we have a growing track record of success in bringing in experts from other fields and getting them to be really committed and productive neuroendocrine tumor investigators. The other really important function of our scientific advisory board is they, of course, lead our research grant review. We use an NIH-based peer review system to evaluate all research grants, and we bring in outside experts when needed, and we also use recusals as appropriate if there are any real or perceived conflicts of interest. And then finally, talking a little bit about our research portfolio. So we were really founded, as has been said over and over today, on the premise that through deep scientific understanding, that's how we will develop better treatments, better cures, and better futures for patients with neuroendocrine cancers. But under that umbrella of scientific understanding, we actually have a very balanced approach. We support research from the very most basic discovery phases of research, looking for new targets for treatments, for biomarkers, all the way through preclinical studies like Diane Reedy-Laguna spoke about earlier, those mouse clinical trials, to even clinical trials in humans. And of course, one of the necessary things for all of this research is the development of models. Models for any researcher out there to have access to study something that they think might be important to neuroendocrine tumors. And of course, having these models will also help young people to decide that neuroendocrine tumor research is a good field to go into, because of course we want people to have the resources that they need to be successful. And then I think just tying this all back to this wonderful Peterson funding opportunity that we have, Ron Hollander spoke about the Accelerator Rfp, these really exciting grants for $1.2 million, these big ideas, the creation of teams with sustained commitments, to neuroendocrine tumors. This will all, of course, be put through a very robust research review, and again, that NIH-based peer review with recusal and use of outside experts. So again, sort of in the vein of the Peterson funding, we've had the wonderful opportunity to expand our collaboration. We've now had a long-standing collaboration, a grant-making partnership actually with the American Association of Cancer Researchers. And now thanks to this funding, we're really going to be able to increase our collaboration with other institutions, both in the neuroendocrine tumor space and beyond. The European Neuroendocrine Tumor Society, we had the opportunity to hear from their treasurer this morning. You know, Ron Hollander and others from NETRF made the trip to the ENETS meeting last year, and you can already see the impact that's had in terms of the response we had to our Accelerator Rfp. We have this wonderful collaboration with INCA. Again, I think the partnerships with NANETS and the ARF have already been spoken about, but just as examples of some of the things we haven't been able to do previously, but we now can thanks to this funding from the Peterson Foundation. A little bit more about this first generation, I guess we'll call it, of Peterson-funded investigators. The first two investigators, Gigi Lozano and Michael German, these are really wonderful investigators that these aren't lifetime neuroendocrine tumor researchers. In fact, Gigi Lozano is really an expert in mouse cancer modeling and has done a lot of work on P53. And then Michael German is actually a diabetes researcher at UCSF. And both of these were researchers that we were able to bring into neuroendocrine tumor research, and now thanks to this funding from the Petersons, they'll become Peterson investigators and be able to continue their important work. What Gigi Lozano is doing at MD Anderson is she's an expert, as I've said, in mouse cancer modeling. She's going to try to create mice with mutations in DAX and ATRX, as these are mutations that our funded researchers have determined could be important in the development of pancreatic neuroendocrine tumors. And really the most important part of this project is once we have these mice, we're sort of hopeful that these mice will develop neuroendocrine tumors, as perverse as that sort of sounds, but once we have these mice that are developing pancreatic neuroendocrine tumors, these are going to be out there in the public so that anyone that wants to study these mice will have access to them. And then Michael German was a diabetes researcher doing some diabetes work at UCSF in California, and he was looking at the pancreatic cells and the insulin-producing cells, and he noticed a really unique phenomenon that when you have these pancreatic cells and they have a particular mutation, which is found in a lot of patients with pancreatic neuroendocrine tumors, and you treat them with a specific type of drug, only those mutated tumor cells die. So from this came this novel idea for a new treatment modality for patients with pancreatic neuroendocrine tumors and perhaps other types of neuroendocrine tumors with the same mutation. So we're really pleased to be able to continue to support Dr. German's work to see if this idea is worthy of preclinical and clinical research. Other than the Peterson investigators, again, we have this partnership with NANETS for our basic translational science investigator. Again, Chris Heafy at Johns Hopkins. It's just such a wonderful story in terms of a breakthrough finding at Johns Hopkins. Our efforts on our own to try to follow up on those mutations, to build models to understand those mutations, to study epigenetics, and then having that new layer of Chris Heafy trying to find cellular models that can again be used to understand those mutations, and really ultimately what those mutations mean for the development of the neuroendocrine tumors, and most importantly, how we can intervene to treat patients with those types of mutations. And then again, we have a picture of Thomas Hope, who was our new ERF NET nuclear medicine grantee at UCSF. For those of you that follow cancer literature, certainly immunotherapy has become a really exciting and productive area of cancer research. In 2013, it was named the Science Breakthrough of the Year, and every cancer meeting really since has really profiled immunotherapy as a real potential game changer for many patients with cancers. And this is because there have been patients with many difficult to treat cancers who are now in lasting remissions. So patients that are not cancer-free for months, but cancer-free for years. So this is really the type of thing we all hope for when we think about trials for patients. And so we're very eager to understand if immunotherapy could be a helpful treatment for neuroendocrine tumor patients. Our portfolio is very balanced with respect to our approach to immunotherapy. On the one hand, we have a bunch of projects to try to understand what does the immune response look like in neuroendocrine tumors. Are patients attempting to mount an immune response, and are the tumors outsmarting the immune system? Is the immune system not able to recognize the tumors? Really trying to learn more about what's going on in terms of an immune response so we can determine which of the many different types of immunotherapy approaches might be the most successful for our patients. We already have 2 grants in this area, to Matt Kulke at the Dana-Farber and also to Tim Meyer at the University College in London. We also have an open funding opportunity. It's actually closed. We have all of the applications, but we'll be doing a review of those applications and hope to have more projects out there soon looking at these tumor microenvironments. And then at the same time, there is one approach to immunotherapy that has been really wonderfully successful, has meant great remissions for many different types of cancers at this point now. And it just so happens that one of the experts in this type of cancer was at the one of the experts in this type of immune treatment was at the University of Pennsylvania, where they also have an expertise in basic neuroendocrine tumor biology, and then of course we've heard from Dr. Metz and expertise in treating neuroendocrine tumor patients. So it seemed just like this perfect opportunity to try to put all these different multidisciplinary areas together and see if this particular immune therapy might work for neuroendocrine tumor patients. So this is the project developing new treatments for NET patients using CAR T cells. I've already mentioned this. It's a collaborative project at the University of Pennsylvania. I'm going to have the opportunity of inviting Dr. Metz to speak more about this project, but this is a project that's involving the use of CAR T cells, which actually works with our Peterson analogy, but these are also frequently known as these serial killer T cells. So this is a really exciting project and one that Dr. Metz is going to speak about more.
Great, thank you very much, Lauren. I'm the only thing between you guys and lunch, right? Okay, so I was asked to talk a little bit around Prrt, immunotherapy, and CAR T cells, and I'm very happy to say that I'm only going to be talking about CAR T cells because the other stuff has been discussed already, and I think we're running a little bit late. One word about Prrt. You saw the data from Dr. Wolin. I think this is a therapy that's going to be arriving across the country in the next number of months, and it really is going to have a tremendous impact on our patients. So a strong supporter of that therapy. Immunotherapy, as you've heard already, has got various kinds, and they're listed over here. Tumor vaccines, I'm not going to be talking about, but that would be a way to vaccinate against the development of subsequent tumors. And there have been some, that has been shown, for example, in cervical cancer to be effective. Cytokines, you've heard already about interferons, interleukins, and other products of immune cells that will reject tumor cells. There's an oncolytic virus trial that is now starting in Uppsala. I have not heard any data on where things have gone with that, but that's a very interesting approach. You give somebody a virus that will infect tumor cells and not other cells. Immune checkpoint inhibitors, you've heard a lot about that already from Dr. Wolin. As you've heard also, we're not sure how these therapies are actually going to work in our particular tumor, although they have been very successful in others like renal cell and melanoma. But I'll be talking about adoptive cell transfer, or CAR T cells, right? So what are CAR T cells? Well, CAR T stands for chimeric antigen receptor T cells. A chimera is an organism that's composed of two or more genetically distinct tissues. So you take the patient's own cell, you transfect that with a virus that changes it, and you turn into a mosaic. The antigen is a substance that produces the production, stimulates the production of antibodies by these cells. And the receptor, as you've heard about already, is a molecular structure on the surface or on the interior of a cell that binds. And in our situation, we're talking about somatostatin receptors. The T cell is a type of white blood cell. There's B cells and T cells. And these are white blood cells that interact with foreign antigens or foreign tumor products to produce an immune response and will ultimately kill off tumor cells. So just a quick word about T cells and B cells. The T cells introduce what we describe as cell-mediated immunity. These are cells that attack foreign antigens. They produce lymphokines and products that have an immune action. They actually switch on B cells and tell the B cells to make antibodies. And most importantly, they have a memory function. So if you can design a T cell that can live in the human as long as any other cells in the body, if tumor ever recurs, the memory function will be reactivated, and therefore you will always be on top of these tumors if they develop again. The B cells, as I've suggested, develop a humoral response. They produce antibodies. They listen to T cells, and that's called antibody-mediated immunity. But we're going to be focusing on T cell and cell-mediated immunity here. So targeted cellular immunotherapy overcomes many of the limitations of traditional chemotherapy. Chemotherapy is like using a nuclear bomb. And other forms of adoptive immunotherapy, sort of like giving somebody an inoculation against a viral infection or against a tumor, is really like a pea shooter. So you want something between the two of these. You want to have a directed therapy that's personalized to the patient. And genetically modifying your own T cells, autologous T cells, can redirect those T cells to recognize antigens that are on the tumor, and then will give you tremendous advantages in therapy. In theory, they'll be specific because that's only against that particular cell. They'll have an amplified response because cells in the body will divide and survive, so they will always be available to respond as necessary. There's a memory, as I've already alluded to. And so as a consequence, if we can really get these CAR T cells to work, it's a smart bomb. Now, CAR T cells were designed originally for the liquid malignancies, and they have now, I think, a standard of care in many liquid malignancies, including chronic lymphocytic leukemia and acute lymphocytic leukemia. And these are what was developed at the University of Pennsylvania by Carl June, who's one of our super scientists. And he chose on the B cell the CD19 molecule, which he decided to target with the patient's own T cells to develop a response and kill off the leukemia of the B cell type. So it works like this. There's your T cell at the top and your tumor cell at the bottom. If you infect the T cell with a viral infection, this is a modified HIV virus, a lentivirus, that will then develop, produce receptors that will migrate up to the surface of the cell. They will recognize this abnormal CD19 molecule that's on the tumor cells and kill the tumor cell. Sounds easy, right? Well, it wasn't as easy as we thought. But the process works like this for leukemia. You start off on the top left-hand corner with leukapheresis. So you take blood out of the patient. You take blood out of the patient, and then you coat those bloods with an antibody-coated bead that recognizes the white cells you're interested. You transduce them by giving the viral infection. You grow them up on a Petri dish. They now have this antigen that they're producing that recognizes the tumor cells. You remove the beads with a very clever process because this is a metallic bead, so you just put a magnet underneath it, the beads all fall out, and now you're left with purified T cells that have been modified to create an antigen that an antibody that recognizes the antigen on the tumor cell, binds to it, and kills them. In the leukemia trials, patients got chemotherapy to make space in the bone marrow, not necessarily in theory if we're going to get this to work in solid malignancies. And so this is being tried now in a number of different tumor types, of which neuroendocrine tumors are just one of those. So I'm going to show you one slide here of the CLL and ALL cells in patients. And here's the vector that was designed, produced this anti-CD19 molecule. And here's an example of a leukemia deposit at baseline that disappeared 83 days later. These are spectacular results. It's essentially become a curable disease. And Carl June, who's the brains behind all of this at University of Pennsylvania, has now developed an enormous enterprise looking at all sorts of different malignancies. So what's the rationale for using this in neuroendocrine tumor cells? So we know that neuroendocrine tumor cells, luckily, have these somatostatin receptors, especially SST type 2, and they can be targeted for this precision type of medicine, so-called theranostics, with or without the G. They're used for diagnosis, for therapy, and for radiotherapy. So the question arises, can SST receptors be leveraged for CAR T cell therapy, just like the CD19 was used for the B cell malignancies? This would require, again, ex vivo, out of the body, expansion of the patient's own autologous T cells that have been engineered to produce this antibody that will then target that receptor and kill off malignant cells without killing off other cells that don't have as much of a receptor density. So our program at Penn has 4 phases, and again, let me mention that this was really driven by the NETRF group. Lauren came to us and said, isn't it funny? On one side of the hallway, you've got Shin, who's our molecular biologist studying multiple endocrine neoplasia type 1 and a neuroendocrine interest. And on the other side of the hallway was Carl June doing all this work in leukemia. And the two, although they knew each other, didn't even know what the two of them were working about until the NETRF came to us and said, hey, guys, let's get collaborative here. So it's really a perfect example of collaboration. So as a consequence of setting up that trial, this is our phases that we're going through with our program. Our first job was to generate and engineer the CAR T cells. The second job was to determine if an in vitro that's outside of the body in a Petri dish, could we show activity? The third part is to wait for these animal models that are on their way and then try this therapy in animal models. We have a mouse model at the University of Pennsylvania for MEN1 tumors, for example. And then once that's all been optimized, we'll start doing trials in patients. So I want to tell you, I get called maybe once a week, can I join your CAR T-cell program? Yes, but it ain't going to be ready for a while. Science takes time, and it is very, very, very hard. It's very hard, it's very expensive, and we have to do this properly. So looking at the first example here is, I'm going to go through a whole lot of slides over here that were lent to me by Shinshin, and hopefully try and describe in the way that I can understand, and I see Dr. Woden looking at his watch, so we'll be quick. This is the somatostatin receptor over here. And the part of the receptor that's outside the body is with this long tail, and our aim is to get an antibody that can get in there and bind. There were 4 of these antibodies that had already been designed and were available, A5, C9, C10, H5. And if you look on this flow cytometry diagram, the difference between those that did and didn't have the antibodies was distinct. So you can separate out those cells and you can purify them and target them. A hybridoma, just to introduce one more term to you, is a process where you inject a specific antigen into a mouse. You collect the antibodies that the mouse makes to that antigen. You then fuse it with a tumor cell and develop a little organoid, a hybridoma, which is made actually out of myeloma cells. And these can multiply indefinitely and sit in a petri dish and can be used to develop this antibody indefinitely that you're then going to use for your science. So here's an example of cultured hybridromas that are shown to release all of these antibodies, A5, C9, C10, and H5, both the heavy chain and the light chain of these antibodies. And so we know now that we can actually do this. We can have cells that will make the antibody. That was step 1, and we thought we were well along our way. So we went on to step 2, and then that is where we started coming against a bit of trouble. We realized that only H5, that's only one of those 4, bound the SST2R recepting cells. We took a THP1 cell, which is a neuroendocrine type cell, that we could then get to express the somatostatin receptor, and we showed that the antibodies actually bind. But you see here the separation between the control cells and the cells that express the somatostatin receptor wasn't that great. We know they bind because there's a difference between the control and the actual transfected cells, but the difference in how well that binds didn't really turn out to be as exciting as we thought it was going to be. And when we actually did some studies looking at how well the uptake resulted in killing of these cells, those were just a CAR T cell alone, that's with the vector transfected, and that's with the antibody being produced. There really isn't any difference at all these different concentrations of both heavy chain and light chain variables. And so it looked great, but unfortunately the antibodies were being made, but they didn't bind and kill cells. So that became a problem. So why is that problem? Well, Shinshin, who's an incredible brain, sat and pondered this probably for weeks and came up with a suggestion that one of the problems with antibodies in humans is they have to get into this little pocket and bind right over there, and that maybe the antibodies we were making were too big to actually find their way into the groove to do the job we wanted them to do. So we went back to the drawing board, and we thought about, are there any other antibodies that we can get to bind domains of small little surface proteins? And he went back to the drawing board, and actually back to Boston, where there's a llama farm, because llamas and camels produce much smaller antibodies than conventional antibodies that you derive out of mice. So you get this very, very small antibody, and you just need to put it in the right place. It was like a small key in a little lock, and we thought, well, we'll try with that. So what did he do? He took these same THP1 cells that were transfected and we knew were producing the receptor. He immunized them into llamas, went through a whole lot of fancy science, and came out with a collection of all this a library of antibodies that these llamas were producing, and then we chose one of those to test back in the system again. And unfortunately, while we do know that the pre-immune serum and the post-immune serum clearly have a big separation on the fax machine, once again, when we actually tried to do the killing specifically, the difference wasn't great. We have taken this further, and we've actually put the THP1 cells with the receptors being expressed with or without the CAR T cells, and there is an increased killing at increasing dilutions, suggesting that we're getting some efficacy. So we've made some progress, but we haven't yet hit that home run. So the future directions for all of this, which has really been tremendous amount of work, I've summarized 2 years of work in 5 slides, is as follows. First of all, we want to optimize the length and or type of linker in these CAR T vectors, because we're very concerned about the actual cell being able to bind and do what it needs to do. We want to screen that library that we've made from the llamas to determine if there's maybe a better example there that's going to be more effective. And what I think is probably a very important process is number 3 on this list, is to actually immunize the llama with neuroendocrine cells that we're going to get from humans rather than THP1 SS2 transfected cells. We might get a better response and it might be more effective. Those are ideas that we're starting to move forward in the next phase of this project. So, just to remind everybody, we've done 1, we're relatively far along the way with 2, but we still got to do a little bit of work on 3 before we get the patients together on level 4. So just to finish off with one last slide, this is enormous amount of work. It has to be done by people who really know what they're doing, and it's a collaborative response. I'm a clinician. I know very little about molecular biology, transfecting cells, and determining levels on fax machines. You really have to have people that know what they're doing. So first of all, I would just want to recognize specifically Shen Hua, who is the Department of Cancer Biology expert at the University of Pennsylvania that I've been working with for many, many years. We have a number of grants together, and thanks to NETRF for that. He's really the brains of the science. As far as the translational research program, this is something that is essential. We're to have Carl June on board, because this is the man who's really developed this whole process, which has now gone countrywide, and many other sites are starting to develop their own CAR T cells. And my own boss, Neil Rusky, in the Division of Gastroenterology, University of Pennsylvania, who supported all of this, as well as the other names listed, and of course, the Neuroendocrine Tumor Research Foundation, who's been able to fund us and make this possible. We're hoping to move forward and really change the paradigm of management for our patients. So thank you very much for your attention.